Let's move on and talk about some particular virulence factors. So I'm going to give you some examples of this. You're working through these in your case studies. As you're researching your pathogen, you're finding out what types of virulence factors does this microorganism have that allows it to get in, stay in and grow, and evade detection from the immune system. So let me give you some examples. We're going to put these in a couple of categories. All right, the first one we're going to talk about are enzymes. Extracellular enzymes are enzymes that are secreted outside the pathogen. All right, so there's a couple of enzymes that work in pairs. Hyaluronidase and collagenase are in pairs. I might draw a little bit on this one. So hyaluronidase and man, I know I'm trying. You guys are giving me grace, right? All right, let me try again. All right, so pen. There we go. Hyaluronidase and collagenase are in a pair, and we're going to talk about coagulase and kinase in a pair. What I need is a pointer. I'll get better, guys. Stick with me, please. All right, so hyaluronidase and collagenase. Hyaluronidase, let's go to, I'm going to go to a picture on this. I would normally draw this on the board, but we know I can't draw it. I don't even draw very well for this. So let me go forward a little bit and show you guys a picture. Okay, so hyaluronidase and collagenase. I know. I'm slow. All right, I appreciate your patience. Okay, so let's think about skin. Skin is made up of tightly packed epithelial cells, right? My ANP people, tightly packed epithelial cells, which is why it's such a good barrier to microorganisms. So microorganisms are here. Our skin is protecting us. We've got several layers of the skin, right? The outermost epithelial layer, and then we have a layer of collagen below that. All right, so this particular bacterium that we see here produces an extracellular enzyme called hyaluronidase. And if you remember from A&P, in between these epithelial cells, these tightly packed cells, is hyaluronic acid. And this is an area that kind of cements these cells together, makes it even tougher for the bacterium to penetrate and get inside. All right, hyaluronidase is an enzyme made by some bacteria that degrades, breaks up this hyaluronic acid. So I have a bacterium out here. It's producing an enzyme called hyaluronidase. It gets secreted outside the bacteria, and then it degrades and breaks up this hyaluronic acid, allowing this bacteria to sneak in between these epithelial cells. There's another enzyme that it makes called collagenase that breaks down the collagen layer. This has allowed this bacteria to get inside and directly past our skin. This is a virulence factor. It's a enzyme that this produces that allows it to invade our cells. All right, there are two other enzymes that we were talking about, coagulase and kinase. If a bacteria gets inside our cell and it's able to produce these enzymes are also virulence factors. What happens is I get a bacterium, it gets inside, it produces coagulase, which, as you can tell from the name, forms a clot. It coagulates and it forms this clot inside your body. All right, so why is the bacteria forming a clot? Well, it's forming a clot, and now the bacterium is inside the clot, and while it's there, time is passing, and that bacterium is reproducing. We go from one or a very small number of bacteria, and inside this clot, it's going to reproduce. Because it's in a clot, it makes it harder for our immune system to detect it. That's the virulence factor. It's evading detection from the immune system. Now, after a while, the bacteria will switch and start producing another enzyme called kinase, and kinase will dissolve a clot. So now, instead of just having a few bacteria, which your immune system can detect and get rid of very quickly, it has been hiding in this clot and produces a whole bunch of bacteria. The kinase releases all that bacteria at one time, and it can quickly overwhelm your immune system. This is another virulence factor. It allows it to avoid detection and elimination from the immune system. All right, so let's go back to that slide. That slide, extracellular enzymes, these are enzymes that serve as virulence factors. All of these enzymes allow for deeper movement into the tissues and avoiding detection from the immune system. All right, so extracellular enzymes are one type of virulence factors some bacteria may have. Some bacteria may have some virulence factors that are actually toxins, something that the bacteria makes that produces inside the host, and the toxin, what it produces, can harm tissues or trigger an immune response. And sometimes our immune response is what's causing the damage. Okay, so toxins, we can classify the toxins in various ways. There are exotoxins. Exotoxins are ones that are secreted by the pathogen. There's also endotoxins, but let's talk about exotoxins first. We can classify that further by what it does or where it works. Cytotoxins kill other cells, so that makes sense. Neurotoxins, you can guess what that does. These are toxins that specifically interfere with nerve cell function. Enterotoxin, entero means gut. So these are toxins that affect our GI system. Now, a lot of times bacteria can get in and infect us, and the bacteria won't trigger an immune response. But once that bacteria starts making these toxins, they will trigger an immune response. So we have antibodies, not necessarily to the bacteria. Remember, a lot of our bacteria, our immune system says that's okay, you can stay. So sometimes our immune system don't kick in until they start making these toxins. So we actually have antibodies that will be produced to these toxins from that. A lot of our vaccines don't actually have the bacteria in it, but these toxins that the bacteria makes. All right, so let's look at this. We're going to look as an example of an exotoxin that can harm us. We're going to look at the disease cholera and look at that. So let's look at that slide. I may have to go for, oh, here it is. So let's look at that slide. These are also epithelial cells. So I have a bigger picture of epithelial cells. These epithelial cells are lining our mucous membranes. So the yellow is my intestinal lumen. So that's the internal part of your gut. I have mucous membranes that line the lumen, and this protects the rest of the body from bacteria that we are eating in our food and digesting in our food, and all the bacteria that's living in our gut. These are protected by this. Again, epithelial cells, so they're tightly packed and don't allow movement through them. All right, so you eat some contaminated food or water. A lot of times cholera is formed in the water. So you drink some contaminated water, and it has this cholera bacteria. This cholera bacteria goes through the stomach, goes into the intestine, has some virulence factors that allow it to get in past the stomach and get into the intestine. Once it's in the intestine, it's hanging out in the intestine, and it will start to produce some new molecules, and these are called toxins. It produces these toxins that are called toxin A and toxin B. Toxins will bind to these epithelial cells lining your mucous membranes, and parts of the toxin will be able to get transported inside these epithelial cells. Now remember, the cholera bacteria are all in your intestine. It's only these molecules that it makes and secretes, these toxins, that are attaching and then entering your epithelial cells. Okay, so now I've got these toxins inside my epithelial cells. A bunch of stuff happens. You don't really need to worry about exactly what happens, but a bunch of stuff happens inside this epithelial cell, and the end result is that your epithelial cells start pumping electrolytes into the intestinal lumen. Salts, chloride, other electrolytes, solutes start being pumped into the intestinal lumen. They are leaving your epithelial cells and going into the intestinal lumen. Now, think about what you know about hypotonic cells, hypertonic cells. If there are more solutes outside the cell, which direction is the water going to go? The water is going to go outside the cell. The water is going to follow these solutes, and the water from the cell is going to go outside and into the lumen, the middle part of your intestine. From that, what's that going to do to your cell? Well, it's being, the cells are shrinking. They're not being able to form a barrier very well. They're not able to do their jobs. We've got a lot of extra water being pumped into this. You're going to get dehydrated. You're going to have problems with this. So people that get sick with cholera, they will have vomiting and diarrhea. All this extra water in the intestine is going to lead to a lot of diarrhea. The cells are going to be dehydrated. They're not going to work. They're not going to be able to get nutrients to what they need. So in the old times, this was a big deal. They didn't have an antibiotic against cholera at that time. They would give the patient more water to try to rehydrate them. They knew they were dehydrated from the diarrhea, and they would try to rehydrate them. Problem was, they didn't realize the cholera bacteria was in the water. So many times they were just giving new bacteria into these patients, and the cycle would start over again with electrolytes being pumped out. So this was a problem. Even if they were getting good, clean water, they were still having issues with patients getting ill from the diarrhea, because giving more water, if I have more electrolytes being pumped out and I give my patient water through their stomach into their intestines, the water is still not getting into the cells. The problem is the electrolyte balance. So now patients with cholera, they're treated with electrolyte therapy, IV therapy, so that we can get more electrolytes into the cells to keep the water from exiting the cells as you're giving more, instead of just giving them water. So that's what's going on. That way the patient gets the water where it needs, instead of just putting more water into the intestine and increasing the diarrhea. Now, eventually, this diarrhea is actually an exit strategy for the cholera bacteria. The cholera bacteria will be flushed out of this host and into the environment. If that infected diarrhea gets transmitted to another host, either gets contaminated in the water supply or caregivers. You're cleaning up a patient with diarrhea, you don't completely wash your hands, and that bacteria gets transferred to another patient. From that, this is how cholera can spread. So many pathogens have an exit strategy, a way to get out from that, because otherwise cholera kills many of its patients and the bacteria will die with it. So a lot of pathogens have some sort of exit strategy. So diarrhea would be an exit strategy for that. Okay, so we can see how the toxins are doing that, how that works. Let me know if you have questions on this one. All right, so going back, these are toxins. This cholera is an example of a toxin and how it can harm us, and that is, these toxins are virulence factors. All right, what else? Exotoxins. Cholera was an example of that. There are some endotoxins. I'm just going to point you back to gram-negative bacteria. Remember, we had gram-negative bacteria had that outer membrane. That outer membrane is made of those lipopolysaccharides. When those lipopolysaccharides are being degraded, if we're killing gram-negative bacteria, those lipopolysaccharides are getting degraded. The lipid A portion of the lipopolysaccharide serves as an endotoxin. When it's released by itself through our bloodstream, then this endotoxin can kill patients. It can lead to blood poisoning, systemic shock, sepsis, and then you get a whole set of signs and symptoms involving that, which can lead to death as well. Okay, so this is some examples of how exotoxins work, and kind of compares those two. All right, so virulence factors. We have enzymes, we have toxins, and the last thing I want to talk about in this section is antiphagocytic factors. There are some things, some traits about bacteria that make it resistant to phagocytosis. Phagocytosis is one of our best ways of protecting ourselves. If bacteria get in, we have cells that will gobble them up, eat them up, right? So antiphagocytic factors include capsules. We've talked about that. Capsules are an outer structure on some bacteria that make it hard for the phagocytes to grab a hold of them. So capsules, if a bacteria has a capsule, it can make it difficult for a phagocyte to do its job. So that is something that is a virulence factor, helps it evade detection from the immune system. There are also chemicals that some bacteria produce that prevent, even if a phagocyte can swallow it up, ingest it, undergo endocytosis, it's supposed to kill it. Some bacteria resist that. Tuberculosis is one. Will actually live inside macrophages that are trying to get rid of the tuberculosis bacteria, and then they will stay inside that macrophage and reproduce and cause problems. So there are some chemicals that are made that are considered antiphagocytic factors. These, again, virulence factors, because they help the bacteria evade detection and elimination from the immune system. Okay. These are our charts. You can go back. We use that one to describe these. You can look at exotoxins and endotoxins and phagocytosis to help you if